our genes too.
Hey y'all, welcome to Farmer Farm. I'm Farmer John. Want to check out my goats?
What?
Goats? What's so special about goats?
They're, transgenic goats, yeah. They were modified by a team of scientists, and now they produce spider silk in their milk somehow. Heck, I have no idea how they did that. Seems like some taboo magic to me. Anyways, that spider silk is really interesting. It's 5 times stronger than steel, and it's more flexible than a rubber band. Also, the spider silk can be used in a variety of items, such as bulletproof clothing. But you can't produce this spider silk by raising spiders in captivity because they fight each other.
Wait, transgenic. You said transgenic. Doesn't that mean genetically modified? So you're saying your goats are genetically modified?
Yep.
Hey, we learned about that in class the other day.
Oh yeah, our teacher told us all about how some animals that are genetically modified can produce human proteins or other proteins. And sometimes they can survive better under special conditions.
How does that work?
A farmyard might seem like an ordinary place, but with genetic engineering you can create transgenic animals to give them novel functions. A transgenic animal has DNA that belongs to another organism inserted into it, so the animal can produce a specific gene product, which in most cases is a protein. For example, Farmer John's goats produce spider silk proteins, which can be purified from the goat's milk. So how does genetic engineering with transgenic animals work? Let's consider Farmer John's goats. First, an egg cell is removed from a female goat and fertilized in vitro, which means in glass. Then, using a very fine-tipped syringe, a DNA fragment containing the gene of interest is injected into the nucleus of the egg cell. Finally, the engineered embryo is implanted into a surrogate goat. About 150 days later, the implanted cell will have developed into a baby goat that can produce the gene product we wanted. To produce the spider silk protein, the gene must be transcribed and translated. Let's take a look at how that works at the cellular level. Transcription is when a segment of DNA is copied into its RNA counterpart, which will form amino acids, the fundamental units of proteins, during translation. Although a cell has many organelles which specialize in different processes, we will look specifically at the nucleus, which is where DNA is located in a cell. During transcription, DNA is unzipped by an enzyme called DNA helicase. Then, another enzyme called RNA polymerase creates a strand of RNA nucleotides that are complementary to the existing DNA. This new transcript is called mRNA, or messenger RNA, and codes for amino acids. In translation, the mRNA runs between a large and a small ribosomal subunit. The ribosome is divided into 3 sites. At site A, transfer RNA, or tRNA, molecules arrive with the amino acid it is carrying. Then, at site P, the tRNA molecule deposits the amino acid and a polypeptide chain begins to form. Finally, at site E, the amino-acyl tRNA bond is broken and the tRNA exits to be recycled. After translation is complete, the polypeptide chains created twist and bond to form a protein. Transcription and translation were both happening at the cellular level. In the bigger picture, the gene product, in this case spider silk protein, can be purified from the goat's milk for Farmer John to use. This method of genetic engineering can be applied to many different organisms.
Oh yeah, that all sounded really interesting. I think I got it now. Hey, can my other animals be genetically modified too?
Yeah, there's already animals that exist like that, like cows that produce insulin for diabetics, or even blood clotting factors for hemophiliacs.
Yeah, and your sheep can be modified to have better quality wool. Genetic engineering can increase the productivity of your farm in so many ways.
Whoa, does that mean my other crops can be genetically modified too? What about me? Can I be genetically modified?
Of course, there's already crops that exist like that, like rice that produce beta-carotene.
And you can be genetically modified. Gene therapy already exists, where people who have disorders relating to one defective gene can have a functional gene inserted instead.
Of course, it's very controversial, and the ethics behind that are a whole other story.